Okay, let's go and get started. So we're going to do codons, which are associated with mRNA. We're going to talk about anticodons, which are associated with tRNA. And we are going to discuss amino acids. How many amino acids are there in nature? Somebody say 100? No. 20. There are 20 amino acids. What are amino acids the monomers of? Proteins. What do you call the bonds that connect one amino acid to another amino acid? Peptide bonds. Okay, good. So let's go ahead and get these defined and do some examples, and I'll let you go off on your own. Codons are 3 nucleotide sequences in mRNA that code for an amino acid. So there were 2 amino acids I've asked you to memorize. What do you call the one that begins with the letter M? Anybody recall? Do you remember? What's it? Methionine. There's a reason I want you all to memorize this one. This is like the train, or the engine to the train. It's the very first amino acid in most proteins. We call it the start amino acid. And every amino acid has a codon to it. Well, the codon for this one is the only codon out of 64 codons I want you to know. What month do we start school? What is the abbreviation for August? AUG. The codon AUG is methionine. It's the only one I want you to memorize. Everything else you'll be able to look at the codon chart. Now, the other one I wanted you to memorize is tryptophan. That was just, we talked about sleepiness and Thanksgiving and melatonin. Well, take a look at this diagram. This is a codon chart. And I will have this projected on the screen while you all are doing your classwork. When you are looking at mRNA, which means you are looking at codons, you first start the A for like AUG. You then look at the U, second letter. You then get the G, third letter. They all intersect right there. Methionine. And notice it says starts in parentheses. Most of them are, or most proteins begin with AUG. If you look up here on the whiteboard, I have in black, I have the normal gene sequence for hemoglobin. Hemoglobin is a protein in your blood that allows your blood to carry oxygen. For all of you, I would assume this is your genetic code for hemoglobin. This is wild type, which means normal, unaffected by mutation. The mRNA codons for that, or the mRNA sequence, is just the exact opposite. So for instance, if the DNA says TAC, the mRNA says AUG. And when you go look at the chart behind me, you figure out what AUG is, methionine. We typically can just abbreviate the amino acid by the first 3 letters. So methionine, valine, proline, leucine, things like that. Now I'll get to all this later. But when you are looking at mRNA, you refer to the charts. So let's go back. But now you need to know what an anticodon is. This is new. An anticodon is a 3 nucleotide sequence, or sequences, in tRNA that are the opposite of a codon. Okay? Now, You guys are my third AP Bio class of the day. There were students in first period that were a little confused with what I am about to say. There were some students in second period that were confused. Fourth period did pretty good. Let's see how you all stand up. It's not a competition, though. I just want to see if how I am saying it is getting better. Maybe I'm not saying it clearly enough. If I have DNA, here, this is an example. If I have DNA that says TAC, thymine, adenine, cytosine, Risa, what would the mRNA complement be for that? AUG. You got it. Hartley, what is the opposite of AUG for RNA? UAC. You got it. That is your tRNA. Guys, this is your codon. This is your anticodon. I'll explain to you what that is in a second. And Riley, what does AUG code for? Methionine. Say it. Methionine. So I'm just going to put MET for methionine. That's what it means. Now, here's the question that was confusing some people, and again, perhaps I'm not saying it clearly enough. When Risa just came, or Riley just came up with methionine, and what I've told you, do you refer to AUG, the mRNA, or do you refer to the UAC, the tRNA? Which one should you refer to when you're trying to figure out the amino acids? That's it. Period. End of story. When you are looking at that chart that I had from your PowerPoint, which I will put up later, you will always 100%, zero exceptions, be referring to the codons, which is mRNA. Okay? Okay. So, let me tell you what the tRNA and the anticodons are all about. Don't write this while I'm writing this. Just watch this and take this in. This is mRNA here. mRNA has now arrived at the ribosome. And written on mRNA is the first codon, AUG. Well, tRNA, which you're going to learn about tomorrow, is going to come into the picture. And if you recall from, I think, last Thursday, mRNA is the instructions. tRNA brings the ingredients. The ingredients to make a protein are amino acids. So a tRNA molecule kind of looks like a cross. But if you look, it is carrying something. This right here is an amino acid. Every individual tRNA molecule is carrying one amino acid. And when you get one amino acid with another, and another, and another, and another, you start to form a chain, kind of like this. Imagine that every little bead on this necklace is an amino acid. You get a chain long enough, you get a protein. You get a bunch of methionines, valines, prolines, tryptophans, a bunch of different amino acids in a proper sequence actually makes a protein. So if that mRNA, which by the way, that's the codon, says AUG, the opposite of that is going to be UAC. That is the anticodon of tRNA. mRNA and tRNA have to work together to get the right amino acid in the right spot. And if that is AUG, then this amino acid is going to be methionine. Okay? So if you guys look up at the whiteboard, once again, the mRNA says AUG. That does mean methionine. It's just the tRNA's job to bring the methionine to the ribosome. And the tRNA speaks anticodon language. The mRNA speaks codon language. They have to be compatible so you actually get the right amino acid. If the mRNA says GUG, that means valine. Well, the tRNA, GUG means CAC. The bottom line is the anticodon is the opposite of the codon, but you always refer to the codon when you're making your amino acids. All right, so I'm going to give you guys a brief little sequence. I'm going to see if you can do this on your own. Consider this one practice before I pass out your classwork. So here's a little practice trial. I want you to come up with the mRNA and the tRNA of the following DNA I'm about to give you, and then once everybody has it, I'll put up the codon chart and see if you can tell me what the proteins would be. Here we go. TAC, GGG, ATA, ACT. Go ahead and transcribe the mRNA and the tRNA. All right. So what you should have is AUG, CCC, UAU, UGA. The tRNA anticodons will be UAC, GGG, AUA, ACU. Are there any questions so far? Okay. Looks like you have 1, 2, 3, 4. No, that's a bad color. Looks like you have 4 codons. So now I'm going to put up the table. I want you to translate. This is a nice little warm-up for tomorrow where you're going to be translating. So I just passed out to you as your homework. Before you get started on that, let's do this. Okay, so I don't have to go back and forth. Help me out. I know the first one is methionine. What is CCC? Jet? What'd you get?
I got proline.
Proline, all right. What is UAU, Lauren? Or Reagan? Tyrene? TYR. And then what is UGA, Ryan? Did you look at the chart that was up on the screen? No. Were you looking at the chart that I had on the screen?
I didn't write down the name, so I got everything else.
Risa?
I got stop.
It says stop, so you don't write anything. That tells you you're done. Let me repeat that. When you see stop, you're done. You don't even write the word stop. You're done. There are 3 stop codons. Okay? So, with that being said, that is the lesson.